Welcome to Gallant Conversations. Gallant is a business-to-business consultancy specializing in industrial innovation.
We drive growth for our clients through strategies and insights in brand management and strategic communications.
If you're in an industry focused on progress at the macro level, we're here to facilitate a conversation, starting now.
All right, so today we're here at Gallant Conversations. We have the privilege to be with Moji over here at Cemvita, a synthetic biology and biotech startup making some serious waves here in Houston and across the world with a global impact. Love for you to first just introduce yourself and give us a little rundown on what you got cooking.
Awesome.
Yeah.
Thanks for the invite.
Yeah.
So Moji, co-founder, CEO at Cemvita, with the other co-founder being Tara, who also happens to be my sister. I come from energy background, and Tara comes from synthetic biology background. And there you have it. That was the idea of bringing these two industries together to create a new category for applications of biotech and SynBio in ways that could transform heavy industries to be sustainable. So we like to say it's like, industry by nature. And the core of what we do is using, waste streams, carbon waste, including CO2, and through biomanufacturing, which is using microbes, turn that into valuable chemicals, fuels that then power everything else.
Yeah, and I mean, it's incredible. And, it's an awesome brother-sister story. So, when y'all were kids, did y'all, have this, mad master plan where you were just going to take over and dominate the world in terms of, industrial materials and feedstocks and all this cool stuff that y'all are working up now, or what?
Absolutely. Yeah, from 3 years old, we had this business model figured out.
For sure.
It just took, no, actually quite the opposite, because I grew up doing physics and mathematics, and Tara did biological sciences.
Yeah.
So I became an engineer. She went into the kind of initially for veterinary medicine, and then for biochemistry and human health. So, and then later I'm going to drilling rigs. She's going to, tissue engineering at hospitals. So we didn't imagine there's anything really in common between these. What brought us together was my last startup experience, which was a company where we were commercializing DNA sequencing in the subsurface, looking at DNA of microbes in the water and rock and oil, and we're building this kind of a 23andMe type maps for the subsurface, so that you could give us an oil sample, we could tell you where this is from. The same way they could say, this guy is from Japan, not from Korea. That was a new data source for better understanding how fluid moves in the subsurface, and companies were using that information to better design their development program. So the main investor in that company was Illumina. Illumina is a really big DNA sequencing company. They're the reason that DNA sequencing became inexpensive, because they built these high throughput machines. So while I was doing this, Tara moved to Houston because she was doing a postdoc with Tulane, but the project was at Texas Heart Institute. And through just organic, dinner, weekend conversations, she learned that this is what I'm doing, and DNA sequencing is part of that. So she taught me more about it, and more I learned about her world of synthetic biology and tissue engineering. We both found this common passion for using SynBio for solving problems in heavy industries, and that became Cemvita.
So you organically came to this conclusion, not synthetically or artificially.
Exactly.
I guess. That's incredible. And so what about the previous startup? That's an interesting bit. I mean, what did you learn from that? Or how did you take those resources and apply it to what you're doing today?
The most important thing I learned was that there is a massive presence of microbes around us, and we're just scratching the surface to understand what their capabilities are.
Yeah.
Most of the history of fermentation microbiology has been with yeast and E. coli. That's just two out of millions and billions of different kind of microbes. So we were seeing that in the previous company, and for any given sample from the subsurface, there was thousands of new microbes that didn't even exist in any database, because no one had gone and take samples, say, from 10,000 feet deep in the Permian Basin and go do DNA sequencing to see what's there, right? So that was when I had this question about, okay, we're sequencing these microbes using the tag as a data source, but what are these microbes? Like, what are they doing here? What are they eating? What are they producing?
Who are their friends?
Yeah, like, are they getting along? How do they change the processes in the subsurface? Because these are natural things, right? We're just observing, and now we're starting to be able to actually make measurements, and then to go a step further to actually control different things. So if, in oil and gas, when people hear microbiology, most likely they think of biocide to kill the microbes. We also have microbes that are problematic, like sulfate-reducing bacteria, because they produce H2S, and H2S corrodes pipes, right? Mm-hmm. So we have a lot of companies and products to kill those microbes. But for every bad microbe like SRBs, there's also good microbes, like the ones that produce natural gas, for example, called methanogens. They generate methane. So it was like, well, who is going to translate these advancements in DNA sequencing and synthetic biology and initial biotech and translate that into new applications for our industry? And I wanted to play that role because someone needed to make the translation. I think actually that's the main reason these things didn't exist before. It wasn't because of necessarily the technology. Technology was there, but people who come from that industry in San Diego and New York, they don't know about oil and gas, and those who are in oil and gas, they don't know about synthetic biology. So that became kind of the area that I really, like they say, double clicked on, and I've stayed there since.
That's cool. And, whenever we're talking about biology or genetic modified, I have to bring up like the CRISPR software. I mean, I know you're probably tired of bringing it up, but I mean, where did CRISPR play with the previous startup and the folks that ended up acquiring you? I'm blanking on the name. You just told me it's Lumia or what have you. Mm-hmm. But I mean, like, was that a precursor to CRISPR, or how does that interrelate with the CRISPR tech? Again, I know it's like buzzwords and stuff, but
no, they're both tools in the same synthetic biology toolbox.
Okay.
DNA sequencing is a starting point in actually knowing what is the genome of a microbe. You probably remember when we did the Human Genome Project, and that took several billions and many years to complete. That's the same thing that Illumina through their machines could do now in half a day for like $200-$300. You could actually do it for your dog too.
Oh yeah. Okay.
So what we're doing, we're saying, let's use this tool for microbes instead of using the common like petri dish. And you have to know what microbes you're looking for and using primers, like very old school methods. Mm-hmm. So we're not changing anything. You're just measuring and understanding what microbes are there through their DNA sequence. CRISPR is now going in and editing certain genes for certain functions. And you have to start with knowing what gene is responsible for what enzyme and what function they have, right? Mm-hmm. Like, say for designer baby example, there's a gene that's responsible for color of your eye. And then you could go edit that for it to, for example, be blue, right? Mm-hmm. So these are different tools in the toolbox of synthetic biology. The way we're using it, though, is quite different. It's more about making the microbes robust to survive in, harsh environments, high pH, low pH. Sure. Acidity, higher temperature, because when you think about heavy industries, when you think about mining, oil and gas, chemicals, that is the environment. So we have to make it more robust beyond the way that they've been used, with like yeast and E. coli in the pharma industry in very clean rooms, like lab coats in a sterilized environment, because that would not scale for heavy industries, right? And that's really the secret sauce of Cemvita. is figuring out those things and like what microbes to use and how to modify them so that they could perform in the intended environment.
Yeah, and it seems like, we engage with y'all pretty on a pretty frequent basis. It's like every day it's just like, oh, y'all are doing something different. Like you're making soap and like perfume and like all this stuff. Like it's not just cosmetics. I don't want to highlight that specifically, but it's just like y'all are continuing just to come up with these awesome, incredible, innovations, if you will, through the synthetic bio space. And, I remember we had a previous conversation about this and, your founding team and like the people that you surround yourself with are heavily involved or were heavily involved with NASA or mission control. I think, at some point, I think at some point you were kind of trying to enter the space applications for your technology with Cemvita and all that bit. And now you're currently going through a fundraise and, you're raising some capital and stuff. So, would you like to kind of touch on that in that transition where you pivoted and went more so oil, gas, and renewable side?
Yeah. So this whole thing is about process of innovation, right? So that's why we experiment these different products, different pathways, different microbes. Sometimes that's the most efficient way to actually go through what doesn't work to get to what does work, instead of just like thinking about it and just interviewing people. You just have to do both. You do it in the lab too. It's the same thing also for the market applications. This is the new space for how synthetic biology is going to help problems. Like you said, one very interesting aspect of that is for long duration space missions, right? Currently, if NASA plans to, or SpaceX for that matter, say go to Mars, for the duration of time that it takes, they take their food with them, right? And it's expensive to take your food with you because it's heavy, right? And you have to use fuel to get there. So that's one aspect. The other one is life support. Like what happens if you run out? And, it's not like a International Space Station where you could just keep sending cargo or like that they're going to come back, right? So because of that, they have this field called ISRU, In Situ Resource Utilization. What does it mean? It's the same thing as circular economy, meaning if you create any kind of a waste, it should not be waste. You should know how to recycle it into something else useful. So for that reason, even, astronauts pee, they recycle it into water, right? So what we're doing in that case was astronauts also breathe out CO2 while they're on that long space mission. And right now that's captured and vented out into space. We thought that's a waste of carbon. Why not use the carbon in that CO2 and turn it into something else useful? And the pathway that we created went to sugar or glucose, because then you could basically add that to water. It's more like a sugary water that has calories in it, right? The point of it is not to make something that's delicious, but it's a life support system so that you have something to survive, basically, right? So that was one aspect of it. The other one was, once you do get to Mars, because 96% of the air is CO2, you don't even have to capture it. It's just readily available. And then if you combine that with the water that does exist underneath the Martian regolith, you could, through a combination of biology and electrochemistry, create hydrogen. And with the CO2, you could make both fuels like methane for the way back, as well as biological molecules like glucose, which you could eat, but also you could use as feedstock for more reactions into a wide range of proteins, vitamins, other life support elements that you need there. So that would be the way to scale life, I think, on Mars, is to use biology in your favor.
Yeah, and like my head's spinning right now because and you spin out like gold hydrogen or what have you, right? Which kind of interconnects with that. And I'm just wondering, and again, I'm not the biologist by any means, but like glucose could also be used as an agent to help with materials as well. Is that right? Like in terms of like you talk about these lunar habitats and Martian habitats, and like they're talking about just like excavating the lunar lava tubes or the lava tubes past volcanic activity to like, that's where you would have kind of life first start and be formed. And I mean, I guess you're saying there's some of these applications, potentially glucose, that's me saying that, not you, could be used as an agent, a bonding agent, if you will. Is that right? For materials and stuff of that nature? Or
it's
no?
definitely sticky because it's sugar.
Yeah.
But I think from glucose, you could also go to cellulose, which is now kind of like cotton.
Yeah.
That's how cotton is made too. So that, I would say, is probably good material for that reason. I actually know that International Space Station and NASA, through their research organization, used to be called CASIS, Center for Advancement of Space or something like that. They actually had a project on this, on how to use the resources that are available to produce a pathway to cotton-like material. And cotton plant is already doing that. And you could create those pathways, from CO2 as well. So as far as building material, I would say that is a good approach. Most of what I have seen, though, is using the kind of the soil in ways that or 3D printing in different ways, as opposed to if you go through biomanufacturing. I think the effort is better utilized is if it's directed towards more like a life support type application. That's really the beauty of glucose, because sugar is the main feedstock for microbes for biomanufacturing. That's how it's done today, even for pharma. So once you have sugar and other microbes, you could make other things, that it may not be feasible to take with you.
Yeah. And I mean, I guess Jared Isaacman in his next Polaris Dawn mission, where he goes halfway to the moon or whatever, he could definitely be, supported through, some of that technology, the life support systems and stuff like that, right?
Yeah.
I mean, that's wild.
What gets wild? There's also application for this for basically perpetual life support. Imagine for survival, if you're going on a mountain or even in the army, if soldiers get stranded or something, right? What do? Someone else will have to deliver food in that case. But if there's a smaller modular of these that could use the CO2 that you're breathing out, as well as the humidity from the air and light, you could replicate photosynthesis to produce glucose, which you could re-inject back into your bloodstream. So you could basically, a human becomes you could just keep going because you have this constant supply of calories.
I guess you would just need to talk to the DOD or something like that and try and get some sort
of behind that would be more what is the other one that is more secretive?
NSA or Homeland Security, CIA.
Within the DOD system, DARPA. Sorry.
DARPA. Yeah.
And then there's
InQtel.
So if they're listening to this, we could talk.
Let's bring some InQtel in on that. Sorry, my phone. I thought I had it freaking on. I broke my rule, man. I thought it was off. So we brought back down to Earth from space, and there's a wide range of applications with what you got going on to help solve a lot of big challenges that we have here on Earth and beyond. And you're talking about harsh environments, and your sister Tara is kind of traveling all across the world, it seems like, or kind of has some certain things in line. And I mean, what about Antarctica? Is that on the docket for you guys? Go on down to Antarctica, maybe go from Argentina, take the little boat over there and see some experiments, or is there anything?
The part of it that will be fascinating is, and Tara knows more about this, is to see what microbes are surviving in that environment. Because I promise you, there's going to be a range of microbes that throughout, years have evolved to survive in lower temperature, not much food available, right? And there's this field, they call it bioprospecting, where you go around the world, especially when you find extreme environments, and you look for microbes and you isolate them, and then you study them and see what machinery is allowing this microbe to survive in hot spring and Yellowstone and in different environments. And once we know that, it's wow, so this is physically possible if you understand the machinery. So then you could replicate that. And this is part of the process that we do. We have our own library of microbes. We call it CemDB. It's like Cemvita database. of microbe, out of which we have already, it's like 150 of them that we've already optimized. So
it's here in Houston, or where
yeah,
is
it's
this?
in Houston. We have
seed
it
vault or
in our lab, and then we have a backup of it also in a
undisclosed location or something.
Undisclosed location that they have really good generators. That if anything happens, we have a backup. That's like our source code, right?
Yeah.
So just think about how much more we don't know of all these microbes that exist in the world. And so that's why bioprospecting is so important, especially in areas that is very rich in biodiversity, or the exact opposite, that is like really hard for microbes to survive. We want to know what machinery is allowing them to do that, and how could we use that to solve specific problems.
Yeah. So, and I use Antarctica as one example because it's a harsh, it's really tough. But like again, hot environments, arid environments, deserts, and stuff like that. It's basically anywhere, right? Like it could be in the middle of a freaking volcano for all we know.
Exactly.
That's cool. Yeah. Well, I mean, this kind of brought me up a thought in terms of like the marketing Gallant side of things, which is like I wonder if like we could just do like a call to like all these little bio prospectors and be like, hey, like. Join us and help us catalog all across the world, do a little freaking trip or something like that. I don't know.
Yeah,
what do we think?
There's some nonprofit through different universities that have initiatives like that. It's the same thing that we do with the species, right?
Yeah.
There's a catalog of different species, and there are people who specialize in insects versus fish or whatever else, birds. So same thing with microbes. It's just like a lot more of them.
Yeah.
And as tools get better, right? Before we didn't have DNA sequencing. So now that we have that, we could learn more and really catalog all of those. Next step is then to understand their capabilities, environment that allows them to thrive, and then see, okay, how do we use that in ways that could also advance humanity? If we take a step back, I would say actually this is what we've already done with other animals. Think about even like farming and stuff, and we've done with plants through agriculture. Next step could be microbes.
Yeah. God, it's really exciting, right? Because you're working on some incredible stuff, and there's a lot of things kind of happening over in the background, and like it can cross-pollinate and really kind of help support a lot of these sustainable development goals, or whatever you have in terms of all the good stuff that's up on the horizon. And I'm just, I kind of want to set it to close. I feel like I could just ask you questions all day, but I don't want to take too much of your time. But, like what do you think the next 5, 10, 15 years, either the industry or your role with Cemvita, like where do you think, I mean, you kind of alluded to it already, but like where you think is like the optimal kind of like, where is the hockey puck going? Or where would you like to see it?
We have, the specific goals in terms of just like execution. So like we're building our first commercial plan next year, and then we want to build 2 more within 3 to 5 year time frame, and then 5 more within the 10-year time frame. So that's a very tangible way to scale what we're doing, to show people what's possible, right? Because then you get more support for it. A lot of these things at the beginning, it sounds crazy. But you know what's crazy? People flying in aluminum tubes around the world. Yeah. That's crazy.
Yeah.
But it used to be that, insane, but then you do it, right? So there's so many of those examples with the jumps in what humanity could do. So for me personally, this is where I'm just doubling down in like showing what biotech could do for sustainability. So that's one part of it as far as Cemvita. Beyond that, I think what is really exciting is 2 things. One is, in what ways AI will help us make jumps. For example, when we start with a microbe, we have to understand all the genetic pathways, what host microbe to use for what a specific reaction. And traditionally, it's being, Tar and other scientists reading, 20 papers and choosing 2-3 different pathways, and then we go do it in the lab to then say, okay, these 2 didn't work, this one worked. Imagine that whole process could be done with high confidence with AI, right, to give us a better starting point. So that could be a big jump in bringing the cost down to get to the answer faster. Yeah. So that's one. The other one is there's a lot of tools that still continue to be developed for synthetic biology, for DNA sequencing, just a toolbox that makes it possible to do biotech. So as these things converge, these are drivers, right? As they converge, they make it possible to go after new applications. Big part of that is our understanding of the natural system. Like gold hydrogen you mentioned is basically turning the subsurface into a bioreactor. So that's cool. New, right?
Yeah.
Because the other bioreactors that we know are on surface in controlled environment. There aren't already thousands of other microbes there. So this new horizon that we're starting to kind of understand, quantify, control, and that's really what is exciting about it. But at the end of the day, it's what is allowing this to happen is that cross-pollination that you mentioned. This is not just one discipline. This is everything. Like you need chemists, you need physicists, you need engineers, you need people from different backgrounds, different industries to bring this together. And guess what? This is how nature also does it anyway. Like there's no physics or chemistry. It just is. Yep. And we define those things to make it simple for us to understand, right? Tara actually wrote a book about this same thing with a Springer, talking about biomimicry and how could we do science differently, where we have more of a holistic view and invite people from different disciplines. And so that's what we've tried to do in our small ecosystem of Cemvita. And go after these big goals, but I think that's what makes it exciting.
And well, before I were to close on that, what's the name of Tara's book?
So, are you ready for it?
Go for it.
It's called Molecular Mechanisms of Autonomy in Biological Systems, Biorelativity of Code, Energy, and Mass. Yeah. And that last part, code, energy, and mass, is why it's called Cemvita, and Vita is like life in Latin. So it's basically saying, how could we learn from nature for transformation of energy and mass together, but is guided by code or DNA information in living systems.
Yeah. So you can grab that like on Amazon.
Yeah, it's pretty much everywhere. If you just type it in Tara, Karimi, Springer, it will pop up.
That's cool.
It's a fun weekend read.
Yeah. I mean, Brandy, principal at Gallant's got me reading on so much stuff, and that's, I think, the next one on my list. So I'm looking forward to it for sure.
Sure.
Again, I'd love to have you another time. I know we kind of have to get going to a photo shoot or what have you. We got you running around all over today, but it's been a pleasure to speak with you, learn a little bit more about Cemvita. I mean, is there anything else that you want to mention in terms of like, where to find you or any kind of announcements you want to have or be on the lookout for in the future?
No, thanks for having me. I think probably the best way is LinkedIn.
For sure.
Just Moji Academy on LinkedIn. And then, we're very excited about what's ahead of us at Cemvita. So we'd love to stay in touch if people follow and as we go and build these commercial plans and see what's possible. Also, like I said, we're always looking for collaborators, especially people who come from radically different industries. They have had a different point of view about the same thing. So very open to those type of conversations as well. So
awesome.
Yeah, don't hesitate to reach out.
For sure. Moji, thank you so much. It's been a pleasure. And
absolutely.
That's a wrap for sure.
Thank you.
Awesome. Thank you.
Yeah.
Thanks for joining this conversation with Gallant.
We hope you enjoyed today's episode and gained meaningful insight.
If you find this entertaining or helpful, subscribe and leave us a review.
And don't forget to share this conversation with your network.
For more resources and to stay updated with Gallant Insights and our amazing client portfolio companies, visit gallantculture.com.
Join us next time as we continue to explore perspectives within the industrial innovation space propelling us forward.
Until then, we ask you to stay thoughtful and curious.